Hello, my name is Moises Claudio. So throughout this term, the genetic disorder that I have been studying and doing vast research on has been on critical congenital heart disease. So to begin, allow me to introduce what this condition is. Critical congenital heart disease, or CCHD for short, is a term that refers to a group of serious heart defects that are present from birth. About 40,000 babies are born with some sort of congenital heart disease each year. Now, the abnormalities that result from CCHD are due to problems with the formation of one or more parts of the heart during the early stages of embryonic development. The problems that arise with the heart's structure cause problems preventing the heart from pumping blood normally to the lungs and the rest of the body. As a result, organs and tissues throughout the body do not receive enough oxygen, which can lead to organ damage and life-threatening conditions. Now, if left untreated, CCHD can lead to shock, coma, and death. Approximately 100 to 200 deaths are due to unrecognized heart disease in newborns each year. So like I said before, CCHD is a group of serious heart defects that are present from birth. There are at least 18 types of CCHD with many more anatomic variations due to the complexity of the heart. Now, some of the most major congenital disorders are hypoplastic left heart syndrome, tetralogy of Fallot, tricuspid valve atresia, and pulmonary valve atresia. All of these disorders are the disorders affect the heart's blood flow through different malformations of specific heart structures. So, starting with hypoplastic left heart syndrome, it consists of malformations to the left ventricle, which can be underdeveloped and too small. So, the mitral valve or the aortic valve and the ascending portion of the aorta may have not formed or are very small. So, additionally, babies with HLHS for short also have an atrial septal defect, which is a hole between the left and right upper chambers of the heart, which are the atria. The tetralogy of Fallot occurs to uh, three things. So, one, the ventricular septal defect, which is a hole in the wall between the two lower chambers of the heart, which are the ventricles. The pulmonary valve in the artery may have an abnormal structure in narrowing, causing something known as pulmonary stenosis. And lastly, the muscular wall of the lower right chamber of the heart, so the right ventricle, is thicker than normal, which is a condition known as ventricular hypertrophy. Um, Moving on to pulmonary atresia. Pulmonary atresia is a defect in the heart involving the pulmonary valve, which takes blood from the heart to the lungs. Now, newborns with pulmonary atresia didn't develop this valve correctly. So, without this valve, blood can go from the right ventricle of the heart out to the lungs. So, in this condition, the heart will bypass and use a different path to get blood to the lungs. Um, in any case, the baby may need surgery or other procedures soon after birth. Tricuspid atresia, like pulmonary atresia, is a defect in the heart involving the tricuspid valve, which takes blood from the right atrium to the right ventricle. The tricuspid atresia occurs when this valve doesn't form at all, and no blood can go from the right atrium throughout the right ventricle, eventually to the lungs to get oxygen. So, in this condition, newborns typically have atrial or ventricular septal defects, which, if you remember, I said earlier, are holes in the walls of the atria or ventricles. So, oxygen poor blood can mix with oxygen rich blood and move across the body. Let's see, on a much deeper scale, there are multiple genes that play a crucial role in the development of the heart and its different structures. For example, we see here four, uh, starting with GJA1. It's a gene found on chromosome 6. It encloses for a protein known as connexin 43, or also known as CX43. CX43 in particular is a protein that is widely expressed in the ventricular myocardium, meaning that it is found abundantly in the cardiac muscle. GAG1 or JAG1 is a gene found on chromosome 20. It is a part of an important gene family that facilitates cell-to-cell communication. Now, it has been demonstrated that JAG1 has been crucial in multiple stages of the development of the heart, including the descending aorta, aortic arch arteries, and the pulmonary arteries. MED13L, found on chromosome 12, is a gene that provides instructions for making a protein that is one piece of a group of proteins known as the mediator complex. Now, researchers believe that as part of the mediator complex, the MED13L protein is involved in many aspects of early development, including the development of the heart. More specifically, the complex in which MED13L is involved plays a role in several chemical signaling pathways within cells. These pathways help direct a broad range of cellular activities such as cell growth, cell movement, and the process by which cells mature to carry out specific functions. Notch 1, found on chromosome 9, is a member of a receptor family that sends signals that are important for normal development of many tissues throughout the body, particularly the heart. These are just four of the genes that are important for the development of the heart. However, if one were to see a mutation, CCHD may often occur. So if we take a closer look at gap junction protein alpha 1, also known as GJA1, we can see how important its role is to the development of the heart, and if a mutation was to happen, how it would affect the newborn. So gap junction protein alpha 1, when not affected by any mutation, is involved in intracellular pathways in the heart. These gap junctions in the heart are involved in pathways for molecular material to pass cell to cell. So this protein also plays a crucial role in the synchronized contraction of the heart and its embryonic development. There has been many studies showing multiple missense mutations that affect the coding sequence of GJA1, resulting in disorders like congenital heart disease. So CCHD is due to its nature and genetic mutations occurs more often than not sporadically. So this means that it occurs in newborns with no history of said conditions. However, close relatives of people with CCHD may have an increased risk of being born with a heart defect compared with people in the general public. So CCHD is inherited in families mainly as an autosomal dominant disorder and is genetically heterogeneous, meaning conditions have many etiologies. For example, parents carrying or not carrying the dominant gene and how they are able to pass it down to their offspring. So in scenario 1, we see both parents are afflicted with a genetic disorder. Since CCHD is mainly inherited as a dominant disorder and each parent is affected, they can either be capital C's or capital C lowercase c. So in this case, they are both capital C lowercase c and give 3/4 of their offspring the disorder as they both carry the dominant gene. However, because they are capital C lowercase c, they have 1/4 chance of having a kid with homozygous recessive genes, meaning not having CCHD. Moving on to scenario 2, one parent is afflicted with a genetic disorder and the second parent does not have a disorder. So the second parent is not considered to be a carrier. In this case, the one parent is afflicted with a genetic disorder and the second parent does not have the disorder and is not a carrier. The offspring will have a 50% chance of gaining the gene causing the disorder. The other 50% have a chance of receiving a homozygous recessive gene, meaning not having CCHD at all. And finally, in scenario 3, neither parent is afflicted with a genetic disorder, nor are they considered carriers. Due to the disorder being dominant in nature, being a carrier would also mean having the disease. So in this scenario, if one parent was homozygous dominant and the other parent was heterozygous, all of the offspring would have the disorder, but half will have the disorder in the heterozygous combination, meaning they will all have a wild-type gene and not having CCHD. We can further see how exactly the offspring is affected if one parent is heterozygous for CCHD and the other parent is homozygous recessive wild-type. So if we look at the pedigree, we see bubble 1 and 2 are both the parents. So the offspring in generation 2 are bubbles 3, 5, 6, and 8. So bubble 5 and 8 are both CCHD positive, showing a genotype of capital C lowercase c. And 3 and 6 both have wild-type homozygous recessive genotypes. So moving down to generation 3, we see bubble 3 had a child because both parents weren't affected, or carriers, their child was neither affected. However, if we look at child 8 and the offspring with bubble 7, we see that the condition is held for generations to follow. So testing newborns for CCHD is not mandatory. However, there is literature that supports testing newborns for critical congenital heart disease as part of the newborn screening test. So babies at the age of 24 hours are tested for CCHD using a simple pulse oximeter test. Due to CCHD causing a lack of oxygen in blood cells, the pulse oximeter test is a great indicator of how much oxygen is in the blood. Another test that may be done is an ultrasound called a fetal echocardiogram, which provides an image of the developing baby's heart. The following algorithm is the steps taken for screening newborns after or before the 24 hours old. Any screening with an oxygen saturation measuring that is greater than or equal to 95% in the right hand or foot, with less than or equal to 3% absolute difference between the right hand or foot, is considered a pass screen, and screening would end. On the contrary, a screen is considered failed if any oxygen saturation measure is less than 90% in the initial screen or in the repeat screens. Oxygen saturation is less than 95% in the right hand and foot on 3 measures, each separated by 1 hour, or if there is a greater than 3% absolute difference existing in oxygen saturation between the right hand and foot on 3 measures, each separated by 1 hour. So as with any test, false positives are possible, although it has been at a minuscule percentage. False positives may lead to unnecessary treatments. However, according to the CDC, to reduce false positive screens, providers can, 1, screen the newborn while he or she is alert. 2, providers can screen the newborn when he or she is at least 24 hours old. And 3, providers can consider further tests to clarify results before taking a plan of action. Infant deaths from critical congenital heart disease decreased more than 33% in 8 states that mandated screening for CCHD using the pulse oximetry test. Screening nationwide could save at least 120 babies each year, according to a new study published in the Journal of the American Medical Association. In conclusion, critical congenital heart disease is if left untreated, can lead to death. However, testing and implementation of screening are being implemented nationwide. Thank you.